Good afternoon. I have the green light from the powers up above to get started. So I have the honor of introducing our colloquium speaker, Dr. Meng-Chuan Lai. Do I get the pronunciation right? So besides being our colloquium speaker this afternoon, Dr. Lai bears many responsibilities and wears many hats. He is an associate professor in the Departments of Psychiatry, Psychology, and Institute of Medical Science at the University of Toronto, an O'Brien Scholar at the Hospital for Sick Children, an honorary visiting fellow at the Autism Research Center in the University of Cambridge, an adjunct associate professor in the Department of Psychiatry of National Taiwan University. So I'm glad that you found some time in your schedule to fit us in. Dr. Lai received an MD from the National Taiwan University and a Ph.D. from the University of Cambridge, where he also conducted postdoctoral research. Dr. Lai's research grapples with a profoundly important question that is very straightforward to state, as you will see, but has proven challenging to answer. And that is, why is it that girls are diagnosed significantly later on the autism spectrum than boys? In pursuing this question, Dr. Lai examines how sex and gender-related factors modulate the presentation, clinical recognition, neurobiology, and etiologies of autism and associated conditions. He is a lead investigator in the province of Ontario Neurodevelopmental Disorders Network, and also in the UK MRC Autism Imaging Multi-Center Study Consortium and the Longitudinal European Autism Project, studying the impact of sex and gender on behavior, cognition, and neurobiology of autism. Among his many honors are the Slivka Ritvo Award for Innovation in Autism Research from INSAR, and the Academic Scholars Award from the University of Toronto. We are absolutely thrilled to have you as our colloquium speaker today. One quick note to the audience members joining us on YouTube, please post your questions. So don't feel shy about posting questions. Post them in the chat window, and I will have the responsibility of reading your questions out loud to the speaker. There is a little delay of about 15 to 20 seconds in the video feed that you will be receiving. So you have to be cognizant and tolerant of that. With that, welcome, Dr. Lai.
Thank you so much, Professor Sinha, and everyone for coming here. And I have to apologize if I look into the camera more than looking into you, because this is the first time I've done in-person talk after 2.5 years since pandemic. So I appreciate everyone being here, and apologies for that. So I was quite intimidated getting the invitation here because of my fragile ego. I had very poor math skills. When I got invitation from MIT, my stereotypes there, like, how dare you speak in front of these people? But the topic is quite interesting, so I'm here to share that with all of you. So let's start with something simple in terms of math that I can handle. So the question about sex and gender and autism mostly start from this reported ratio of diagnosis, and you will read it from the textbook, and lots of articles citing the diagnosis rate is about 4 to 5 to 1 male to female. This is a meta-analysis conducted by Will Mandy's group in UCL, and they basically stratified those epidemiological studies into studies actively ascertaining people, meaning that they go into the community, do screening, and conduct so-called gold standard diagnostic assessment. And they also compare those rates to the rates of diagnosis in existing medical or educational databases. Then they see that there is an attenuated rate, male to female rate, of 3 point something, 3.25 to 1 in this case, in the active ascertainment ones. Assuming the diagnostic instruments themselves are unbiased, this finding tells us two things. First, there is still a male preponderance in epidemiological studies, suggesting that there are some etiological factors associated with sex and gender in terms of autism. Secondly, there are some ascertainment biases ongoing, so people are not picked up in the community. The question is actually not very straightforward or simple to answer because most studies have only one variable of sex/gender. It's not well defined in many cases. It's confounded, it's complex. If you want to get into mechanisms about sex and gender to explain why these are happening, then you get into this question of what do we mean by sex and what do we mean by gender. So as we know, sex and gender are both like umbrella terms containing lots of constructs. So if you're interested into mechanisms about etiology, for example, you might be interested into sex chromosomes or the X chromosome activation or sex hormone effect, whereas if you're into questions surrounding diagnostic bias or behavior presentation, you might be talking about personal gender, so like internalized gender roles, or you might be talking about other gender-related aspects, including stereotypes. So there are multiple constructs that needs to be clarified for future research, but everything I'm going to present later actually may not have all these constructs. It's still like one variable of sex and gender. And why is this important? Because sex and gender-related components actually speaks to different aspects related to autism and other neurodevelopmental conditions. For example, on top of that, they can be related to the emergence of the condition, of the role of moderating the etiologies or mediating the occurrence of the condition. They can also have impact on how phenotypic presentations are modulated and also how things are picked up in the community. And finally, all these are related to how service delivery can be changed for the future. So I always have this ABCD to remind myself how important they are. A stands for etiology in British English. B and C stands for biological heterogeneity and also clinical care, and then the delivery of care. So let me start with the clinical and clinical care delivery aspects. This is a systematic review conducted by Frankie Heppe's group in King's College London a couple of years back, basically asking the question that Professor Sinha raised: why are girls and women being picked up being autistic at later ages compared to boys and men? And they summarized the literature to say that some are related to how an individual presented their characteristics, may be related to the autism dyads or other co-occurring conditions, but others are related to what they call perceived barriers, related to how things are recognized by other people. So I summarize that into what we call contextual factor versus individual factors. Let's start with the contextual one. So this factor can be related to how sex, and mostly I would argue gender-related factor, affecting how things are interpreted in the lens of parents, teachers, caregivers, or clinicians. So this is a nice study to start with. So this is from the TEDS study, which is a general population-based twin cohort study done in the UK. And the study by Dortinski et al. From Frankie Heppe's group showed that if you compare those people who have high autistic traits without an autism diagnosis to those who have autistic traits, high autistic traits plus a clinical diagnosis in girls, those who have a clinical diagnosis tend to have higher proportion, have concurrent cognitive or behavior challenges. That difference is actually less prominent for the male counterparts. Although they're a small sample, this speaks to the possibility that girls need to present more for them to be diagnosed clinically. That's almost like replicated in different ways in the clinical consecutive referrals in the Dutch dataset, showing that for those people who come into clinical assessment, who eventually get ASD diagnosis, the girls compared to the girls who have diagnosis compared to those who do not have a diagnosis, they have elevated CBCL scores, which is parent-reported emotional behavior traits. That level of differences is not evident for the boys group. The most prominent example for that is probably this one from the Children's National Group. What they have is a big research clinical cohort. They matched autistic boys and girls on ADOS scores, meaning that they present similarly in front of people. Now, they asked the parents to rate their SRS scores, and the girls are all scoring higher. The parents also gave an interview for Vineland, which is adaptive functioning scores, and the girls are actually scoring lower, suggesting that given the same presentation of autistic characteristics in front of the clinicians, girls tend to have more traits recognized by the parents and poorer adaptive functioning skills. So this one study is led by, again, Will Mendy's group, and we looked at how likely are the recognition bias playing a role. So Alana Woodcock created this, what we call vignettes of description of autism, presented to primary school educators in the UK. It was called female phenotype versus male phenotype, which I will argue later is probably not the good term. It's more like typical phenotype versus more nuanced phenotype. And then this is a vignette presented with those two different presentations, but another factor of manipulation is the gender of the child implied by the name. So those vignettes are presented to primary school teachers, asking them how likely you think this child has an autism diagnosis, so they should be further referred for assessment. You can see that if they are presented with the typical phenotype, the child's gender doesn't matter. If they are presented with the more nuanced phenotype, then it's more likely for a boy to be referred for assessment and having autism in the teacher's mind compared to a girl's name. So that's a very nice illustration that when things become more muddy, more nuanced, the gender of the child, the perceived gender of the child, does matter. So for this part, I like to use this quote to summarize. This is from a quote from a qualitative study of ours. It's from a teenage girl who had her diagnosis at about 11, and she spoke, actually she revealed this to her dad, and her dad revealed this to us. I think this really nicely summarizes what they meant by contextual bias or recognition bias. Now, if we turn to the individual level factors, then what should be recognized is sex and gender-related factors can actually possibly modulate how a presentation of autism looks like, and also their developmental trajectories. So this is almost a known knowledge in the field that if you use these conventional measures, standardized for decades, ADR, ADOS, CARS, SRS, for those who have been clinically recognized or they meeting clinical criteria, if you compare at the group level, the social communication scores may not differ too much, but girls tend to be scored having like fewer repetitive behaviors. And just to be more nuanced, it's actually usually less stereotypy and but at the same time, also less fixated interest being scored or picked up by the clinicians or the researchers. But that level of differences doesn't really imply for these analyses to say you need a different threshold for using this questionnaire. Downside being that these are studies with case control design. So you have those who have already been diagnosed or meeting clinical criteria versus those who do not. And you just don't know how these things perform in those like gray zone areas. And that's a study that hasn't really been conducted well. Another interesting finding I'll refer to later is that across this large-scale data set, parent-reported social communication difficulties for autistic people tend to be higher in girls in teenage compared to boys. And that's an interesting phenomenon that we'll have to turn to later. Now, this is the most important finding maybe for conventional measures. But there may be other things that's not picked up well by the current measure or the current scoring of these measures. And one, I will raise two examples here. The first example is what we mean by social attention. I always refer to my friend Claire Harrop's work in North Carolina. Claire used eye tracking to observe the child's social attention approximated by their fixation time to the video, especially the face of the children playing. And they are like a socially lean and socially rich play session. As you can see, they recruited autistic girls, boys, school age, and non-autistic girls and boys. Now, if you use this attention to face area as a proxy for social attention, especially in socially rich phenomenon, you see there's a cascade effect So there's a sex/gender effect in typically developing group. So typically developing girls pay more attention than typically developing boys, followed by autistic girls, followed by autistic boys, suggesting that it's not only autism diagnosis playing a role, the gender and the sex of the child, actually we don't know whether it's gender or sex, but it's mixed effect, also play a role, and you have this cascade effect. This is almost replicated in our LEAP dataset led by Teresa Del Bianco, just published recently. Again, in one of the stimuli, which is dynamic social scene, you see that cascade effect for highest attention for typically developing girls and women. This is age 6 to 30, followed by typically developing men, boys or men, followed by autistic girls, women, followed by autistic boys and men. So another example is what people nowadays use the term camouflaging, masking, passing as non-autistic, referring to social coping mechanisms. And that's probably best demonstrated by playground behavior. So this is Michelle Dean's work from Connie Cassar's group a while ago, basically recording what's happening in the school playground for autistic boys and girls and non-autistic boys and girls. They went into the playground, analyzing their play behavior, classifying them by whether they are solitary, their joint engagement, or they're actually interacting in structured games. So not surprisingly, but sadly, autistic boys and girls spend most time compared to the non-autistic ones in solitary activities. But what is interesting specifically here is that autistic girls actually spend quite a bit of time what they call joint engagement activities, meaning that they're just around the group of people. Whether they're interacting in depth is unknown, but they're around there. And that's actually to the level similar to typical developing boys, which suggested that if you just look afar, you see they are there, they're in a group, they're not alone. So some of them may actually be having difficulties socially, but you just don't know, but they're just there. And this effect can be driven from the gender landscape, because you might argue that girls tend just to have a group together and play together in that age range, or it could be the strategy the child adapted, which we don't know so far. Another good example is actually this task, which is a behavior reciprocity test. It's like Donald Winnicott's squiggle. So basically, you draw together, the child draw together with the researcher, and then they analyze the reciprocity of the drawing together. The interesting part for this study is that boys and girls, autistic and non-autistic, 4 groups, the autistic boys and girls are matched on their theory of mind measurement and also their ADR scores. However, behaviorally, autistic girls actually show similar level of reciprocity compared to non-autistic girls, whereas autistic boys show lower level. So even given some similarity in measures in theory of mind or autistic symptomatology, they behaviorally act more reciprocal. So that's in a way characterized as what we call masking, compensation, or camouflaging, or social coping. We work together with Laura Hall and in Will Mendy's group, trying to learn this from adults' perspective. And as some of you might know, there's an instrument created based on qualitative research called Camouflaging Autistic Traits Questionnaire, which is essentially a questionnaire coming from qualitative studies with adult autistic individuals to characterize the strategies they use in coping with social situations. And there's some factor analysis showing that the strategies can be separated into compensating for things that they do not have naturally or intuitively, masking autistic features, for example, suppressing mannerisms or stimming behaviors, and the motivation to assimilate to the group. This is just one way of categorizing or characterizing what this looks like, but from adult literature. This hasn't really been applied very well in the teenage literature. I mean, it's been used, but not really well validated. And we actually don't know much about the children population in terms of how these can be perceived and reported. But this is a tool that has been used to characterize the intention to fit in. We also use another way, which is basically operationalizing camouflaging by secondary data analysis to say, what are the discrepancy between the outward autistic characterization, in which case ADL score, and these internal autism status, which we defined as theory of mind score, as well as self-perceived autistic traits. And this is actually an immature way, and it's not the gold standard. I know people use it, but we're just trying to explore what it looks like. So we create a score on that discrepancy, interestingly showing that in an adult sample, autistic women tend to have higher scores of that compared to autistic men, also interestingly, we happen to have fMRI paradigm, which is involving self-other processing. So as you can see here in MPFC, which is the area that's responding usually to self-other processing. So in this sample, we have a significant diagnosis by sex interaction, meaning that autistic individuals have hypoactivation compared to non-autistic men, autistic men compared to non-autistic men, but there's a hyperactivation in this sample in autistic women compared to non-autistic women. What is interesting here is that that discrepancy measure that we created somehow is quite associated with the level of neuroactivation here. So again, this is very indirect cross-sectional analysis. We don't know where it comes from. It could be, a chicken and egg problem, but there is some association maybe implying there is a neurological or even cognitive basis of these conceptualization. But it is important to note that what we meant by camouflaging or described by camouflaging from lived experiences is not probably exclusively an autistic experience, because it actually fits into impression management. If we go back to Irving Goffman, we think about how we present ourselves to each other, to you, like I'm performing in front of you, and this is different from when I'm with my family, a little bit. However, this is the ubiquitous human experiences. We all manage impression for a reason of, we want to have some enhancement in social status, or we are actually compelled to do so. And it also involves lots of cognitive nature that's maybe shared by lots of people. And also there will be consequences of that, positive or negative. But it could be some characteristics of neurodivergence, autistic being one, it could be others, that's make this experience unique. So for example, how compelled you are to perform socially if you have that motivation to do so. How difficult would you be to constantly shift from self and other perspective in order to achieve the goal of simulating and then, all the way like compensating or masking, and how laborious would that be, and how that consequence may look like for you if you are neurodivergent. So I think we're trying to link this back to more ubiquitous human experience to say that, well, it is important. It's not diagnostic for autism. It is important. Autistic experiences for some people. It's also ubiquitous for human beings socially, and we can really leverage a lot of social psychology, social neuroscience understanding to better understand this aspect. Now, developmentally, sex and gender can also play important roles. I want to draw attention to this Baby Siblings Research Consortium dataset publication. Essentially, as you might know, these are babies who are tracked from birth, who already have an older sibling who have an autism diagnosis. Now, they measure their Mullen scales, so early developmental scales from 0 to 3. By age 3, as you can see, males are in the black line and females are in the red lines, and those solid lines are those who have a diagnosis of autism by age of 3 already confirmed in this high probability sample. Females are actually performing better across all domains compared to males. And this is in contrast to what we have learned from the past literature, that it's always more cognitively impaired or neurologically challenged girls who have an autism diagnosis. I would argue that might actually be a bit of an ascertainment bias, because if you track people from this particular cohort, from very early on, you pick up things, you actually see a better developmental trajectory, even for those who have a diagnosis at the age of 3. The similar situation has been founded by other cohort who included people are diagnosed early in life, so not only later in life. So the pathways in autism in ASD study is led by Peter Zamari, my mentor. It's an inception cohort for those who are diagnosed with autism age 2 to 4 in Canada. They track them over the years. Now they stratify them based on longitudinal trajectories by ADOS scores. So across the cohort, there are 2 groups, one with declining ADOS scores, the other with high sustained ADOS scores. Girls are enriched in this declining group. And this finding is similarly presented in a smaller cohort from UC Davis Autism Phenom Project. Again, tracking ADOS scores for those who are diagnosed early in life. You have a declining group. The declining group is enriched with girls. Not all exclusively girls, but enriched with girls. So even those who are diagnosed early in life, you do have a trajectory of some showing less, at least ADOS scores, over time. Now, what that means is unknown, right? Does it mean that this makes this person less autistic? Well, we don't know. Or is it because behaviors are shaped over time? then it becomes less obvious because of learning or other trajectories. We don't know. It could be gender effect, socialization effect, it could be sex effect, and we just don't have the data to speak to that yet. Another interesting baby sibling study is from the IBS network, and they recently published a study, again, using data-driven longitudinal trajectories to identify subgroups. On the y-axis, these are the scores on the social communication domains or RBI domains. And you can see that they are separated in two groups. One is like with high scores and then gradually declining, and the other is lower scores. The female groups are usually having less scores compared to the male groups. But what is interesting is if you look at this Venn diagram, so the percentage of those, again, these are infant siblings with, within high probability of having autism to start with, because there's already an older sibling with autism diagnosis. The sex and gender spread in these two groups, in a high trait or low trace groups, are actually closer to one-on-one, and across both social and RBI domains. But with careful clinical assessment, those girls are actually, the proportion of those girls getting a clinical diagnosis at age of 3 is still lower than the boys. Now, this is a room for interpretation, what this actually means. Is it like they have less clinical need despite the high presentation, or is it related to some contextual bias? We don't know. But I think there are like a lot of clinical, contextual developmental issues that needs to be resolved. One small thing which is important but really not well studied so far is the intersectionality, well, at least within neurodiversity and gender and sexual diversity. So one study that we've tried to do is led by Varun Warrior, which looked at the rates of reported autism diagnosis in large-scale data sets, and it's not a very well-characterized gender diverse or trans population, because it's just one question asking what's your identity, and that's asked not in a very consistent ways across studies, but they do characterize non-cisgender men, non-cisgender women population. And in that population, there's a heightened probability for them to report having an autism diagnosis. Not only autism, actually, also other neurodevelopmental diagnoses like ADHD or LD, as well as psychiatric conditions. And there's actually enough study for meta-analysis to be done, just published recently, looking at the rates of autism diagnosis in gender diverse, gender incongruent people, giving about like 11% of prevalence. And as you might know, this is much higher than what you'd expect in the general population. Even in the CDC recent number, you would say like 2%. This is still much higher. So definitely the co-occurrence is there. They're much less from the autistic population. So like, what's the prevalence rate of gender diversity in the autistic population is less well studied. This is a study from the Netherlands Autism Register. They looked at self-reported identified gender. And you can see that for the birth-assigned male group, actually there's about like 8 to 9% of them reporting non-cisgender. Again, like identified gender. It's not worded exactly that way, but you can see that's non-cisgender identification. And it's actually much more for birth-assigned female, and these are all autistic people. We're trying to ask the question, is this diagnosis specific or not? So we looked at the Province of Ontario Neurodevelopmental Disorders Network, and this is led by my PhD student, Kelly Moo. We use a range of categorical diagnosis within neurodevelopmental domains and dimensional traits to predict a score from gender identity questionnaire for children. So parent report of child's behavior, and we score that in a variety of ways, but this is from a concurrence, incongruence, like very overall generic perspective. Now what we see is that categorical autism and ADHD diagnosis no longer becomes the predictor for gender incongruence reported on this GIQC from the parent. Instead, is the SCQ, social communication subscale, that predicts the gender incongruence of the children at the time when they fill out the survey. Now, this is interesting in a way because it's almost like an imperfect longitudinal analysis, because SCQ, social communication score, asks about 4 to 5-year-old, if you remember. So there's like some recalled child characteristics at 4 to 5 predicting child's gender incongruence reported by the parents. Downside being, in this data set, we actually have very little self-identified non-cisgender people. And we suspect there's lots of discrepancy between parent report and self-report. But that's the imperfect data set that we have. So what does this all lead us to? I think it helps us to understand how does the concept of autism actually morph over time. And there's a lot of discussion in the field about whether we should go back to prototype autism versus like more extended type of autism. The broader autism phenotype ideas has been there for a long while. Where do we draw the boundary? And there's also this idea of like female autism phenotype that's come out in the past few years, which I'm guilty of using that term a while ago, but I'm arguing that may not be a good term because it's not specifically like female, male thing. It's something that we learned from people who have more nuanced presentation, and most of them actually in this case, girls and women and gender diverse individuals, that actually tells a bit more about how autism presentation can manifest depending on how they are modulated by sex and gender. So I think nuanced phenotype would be a better word or idea to think about it. It's not sex and gender dependent, it's sex and gender modulated. We still need to know what they actually mean. And I'm just giving a bit of example here about, if we trying to highlight a few points based on the DSM criteria, what that might look like. So, for example, we know there's heightened social attention for, autistic growth. So, it's not like there's no attention to social situations. Social awareness could be there. Conversation, can be learned to be, seemingly reciprocal, but it could be well rehearsed. And for example, things like social relationships, there's definitely not true that autistic people do not want friends. I mean, we have lots of other studies showing that autistic people actually long for friendships, at least a majority of autistic people, but find it hard to manage or cope. So, there are more nuances that needs to be understood, taking the DSM framework, for example. And this is just one paper that we put out there trying to highlight a few points. There are good books that's going to come out from experienced clinicians, I'm thinking next year, that actually will address issues like this, like how do we operationalize the DSM criteria in a way that's clinically meaningful, informed by lived experiences and research. And one final little word before I move on is this idea of this camouflaging impression management. It actually fits with what people are trying to say in DSM criteria C, which is like it should be there from early on, but it may not manifest clearly until decompensation or context demand changes over time. And I would argue that there must be something there for one to camouflage. Right? I mean, if there's nothing there, then it's really hard to argue this is. So the question is, how do we understand what that is? So then there must be something in early childhood for one to start to cope with lots of effort and with lots of practice. It gets better over time, but it might still be challenging, even with challenging at the time of social interaction or during a prolonged social interaction. So lots of clinical assessment for late diagnosis is not like a one-off thing. It's not like one-off ADOS giving diagnosis. You might actually have to observe how things evolve over time across different instances, getting multiple collaterals to achieve an accurate clinical impression. Sorry about that. That's a lot, but I'm going to run through a few other things if you allow me to. So. Moving away from the clinical aspect, the other key idea is about how sex and gender play a role in terms of biological heterogeneity. I'm using a few neuroimaging studies to make the point. This is led by Dori Flores and Jose Vilho in Adriana DiMartino's lab, and we all collaborate on this huge work. This is like typical way how people trying to understand sex-dependent effect. So you take a brain imaging data, in this case, resting state function MRI metrics. We ask the question, are there sex-independent autism-related characteristics? Are there sex-dependent autism-related characteristics? And as you might know, you just run a mass univariate model. You look at where is the sex by diagnosis interaction, which implies in that particular area, you get that sex-dependent effect. So that's localization. And what Dori found here was that in the dorsolateral occipital cortex, in voxel-mirrored homotopic connectivity, you get this sex differential diagnostic effect, which is somehow actually correlated with behavior metrics and some, you have some brain behavior correlation. So that's what people usually do and trying to understand. So it's a very important way of understanding localization and whether there are sex-dependent, domain-specific or region-specific effects. But that's not good enough. So what we're trying to do also is to think about what if the effect is not manifesting in one particular region or particular network, but what does it look like globally? And that's really coming from some neuroanatomical studies showing that, well, the autism effect in men and women, or males and females, actually manifests in different brain regions. So let's take a simple example. I think there are two possible univariate examples of sex modulation effect. One is what we call quantitative sex modulation. So the idea is quite simple, coming from this, like, you need more for you to become clinically autistic. So if you need if you need a 1 point change for you to, from neurotypical to be neurodivergent, you need a 3 point change at the same variable for you to become neurodivergent. So that's a quantitative modulation. For females, you need more. It could be the other direction, which is that you actually have different changes for you to become clinically autistic. So that's what we call qualitative sex modulation. And this qualitative example is usually what people find for the localization interaction effect, because that's like this ordinal interaction. That's usually how people are powered to find. So we took on a data set from the province of Ontario Neurodevelopmental Disorder Network plus a Hospital for Sick Children network. We construed two assessments or two statistical tests addressing these two examples. So one on the left is what we call local magnitude model, essentially asking the question, if you see an effect happening related to autism neurotypical differences, do you always see a higher effect size for girls compared to boys, or female compared to male? If that's true, more than random, that's evidence for global quantitative sex modulation. On the other hand, we simply compare the spatial correlation of the diagnostic effect by males and females and to see how much they correlate. So again, if they are more dissimilar than you would expect by random, they are evidence for qualitative sex modulation. We don't find any evidence, actually, surprisingly, in neuroanatomical metrics for the quantitative sex modulation part. We do have evidence in cortical curvature and also in subcortical volume that showed no matter how you threshold it, you do have more dissimilarity, basically in this case represented by an undershooting of the observed spatial correlation coefficient compared to the null distribution by permutation, and between the diagnostic effect in males versus females. And then if you look at the voxels the vertices contributing to that dissimilarity, it's actually clustered around across the brain, but not like focal, like localized regions. So it's actually a quite diverse, but a clustered, widespread regions contributing to that. So we found basically in this case more evidence for qualitative sex modulation or sex dissimilarity given the same clinical diagnosis compared to quantitative sex modulation. And that is somehow also found in different samples. In this case, for example, this is in the rat sample, which is led by Sven Bolt in Sweden, and they have a general population sample, twin sample, also enriched with autism diagnosis. What they found is that for 1 unit of increase of SR score, you actually get more regions associated in female compared to males, and it's more widespread. So the assumption, the interpretation could be that, yeah, you get more changes in the brain for you to have 1 unit increase in SRS scores. However, there are also different regions associated. So it's not only more changes, it's also more widespread changes for that to happen. A recent publication from Joshua Lee from Christine Unorda's group, again in UC Davis, showed that they analyzed the multivariate effect size that differentiated autism and typical developing children, toddlers, on the amygdala-connected networks. Now, they also get the regions that contribute the most to the autism control differences. And as you can see, those regions contributing the most to the male sample is in these green bars, and those contributing the most to the girls or the female sample is in the pink bar. And they don't actually overlap. The Dye's similarity coefficient is actually quite low. And that also applies similarly to the diagnosis by age interaction effect, which it basically says that the regions contributing to the developmental trajectory changes dependent on diagnostic status also is quite different in the two sexes. So that speaks to the possibility that actually the neurobiological basis associated with autism is quite dependent on the sex of the individual. Now, finally, as I mentioned earlier, sex can be an angle, and gender is also an angle, but there's very few studies on that so far, for heterogeneity. But it also has this interesting implication of what does it speak to etiological relevance. So as you might know, one big thing that people talk about is the genetic differences associated with autism. And one big thing which was termed in the past female protective effect, or higher probability for males to be diagnosed with autism, is related to genetic findings. And for example, this is the most recent updated data set from Jonathan Sebast's lab in UCSD. They look at the SPARC and SSC sample, but also a local UCSD data set, and basically replicated the fact that in a group level, autistic females tend to carry higher load of rare de novo variants compared to autistic males. But they also find that the common variance load is also higher on average for autistic female compared to autistic males. Varun Warrior in Cambridge and Simon Baron-Cohen's lab also look at SPARC and SSC's dataset, and basically, again, demonstrating that for those rare variants that's enriched in females, they're more likely to be those associated with developmental disability de novo variants, but not so much for those associated with non-developmental disability associated rare variants. But what's even more interesting here is that if they divide the sample by the IQ range, you can see here on the y-axis is the load of polygenic score of common variants that tend to be higher in women or girls who do not have intellectual disability, and even higher for those who have IQ in the average range. So it seems to be the case that the genetic associations are related to both rare and common variants. And this is a work, again, replicated from Elise Robinson's lab here in Broad Institute, showing that if you stratify people by whether they have de novo mutations to start with, those who have no de novo mutations, who are female assigned at birth, has the highest polygenic score of common variants compared to those who especially compared to those who have rare variants and those who are male assigned at birth. So again, suggesting that rare variants and common variants seem to play a role together in contributing to the probability of one becoming autistic. Now, the question is, we know there's this genetic evidence, but what are the mechanisms that mediated or modulated that genetic influence? So that brings us to the final aspect of my talk, which is we might learn from sex differentiation mechanisms, and also gender socialization, but that's actually less studied so far, of knowing what those relevance are. And one key point is this idea of convergence. So I'm citing this fascinating work from Peg McCarthy, who, as you might know, is an animal researcher about sex differences and especially developing brain in rodents. So Peg McCarthy's group basically identified that in mice, in rodents, you have this sexually dimorphic nuclei, which is differential, actually dimorphic in mice in terms of size. But what is also dimorphic is not only the size, but also the circulating microglia status. So in male mice, the circulating microglia tend to be more active compared to the female mice. Now, this process is masculinized by estradiol. As you might know, this is a sex hormone, and in mice, that's the active ingredient. It's also mediated by the immune pathway. It's PGE2 mediated pathway. So in the end, what it implies is that the masculinization, quote unquote, process in mice is actually mediated also by immune mechanisms. So the two mechanisms actually converge. So that proposal leads to this idea of a typical sex differentiation process placed male animals, at least in rodents, at a heightened immunological status or inflammatory status. So you have other perturbances into the immunological status prenatally that actually may contribute to the possibility of neurodevelopmental outcomes. And as we know that inflammation or maternal inflammation in general is itself a possible factor for neurodevelopmental outcomes to start with. So that's where it, from a mechanism level, put it together. in a human epidemiological data set, there are some meta-analysis showing that maternal polycystic ovary syndrome is associated with the possibility of the offspring being diagnosed with autism, but also ADHD. And it's the same actually across male and female children. It's not only ASD or autism ADHD, but also chronic tic disorder. So PCOS itself may be associated with later offspring diagnosis of neurodevelopmental conditions. Now, there's confounders for sure, because although PCOS is a hyperandrogenic status of the mom, it's also associated with obesity, metabolic issues, immunological issues, or medication use. So you really cannot tease these apart from the effect to offspring outcome. But this is something to start with that people talk about in the epidemiological data set. Another line of evidence comes from Simon Baron-Cohen's lab, looking at the Danish historical birth cohort and asking the association in the prenatal amniocentesis-derived hormonal status and offspring autism diagnosis. And these are exclusively in male children, male assigned at birth, because they just don't have enough sample for females. So what they found is that if they look at what they thought related to the delta-4 sex steroid pathway and cortisol, they're all related. And altogether, if you use a PCA to derive them, and the first PC represents the most variances of the effect of these, those autistic children actually had a higher, associated with higher prenatal steroid-related hormones here. Interestingly, it's not only these androgens or cortisol, because they actually had a follow-up analysis just published 2 years ago looking at the estradiol or estrogen molecules, and they all are associated as well. So it's not as a straightforward thing of masculizing versus feminizing or sex hormone itself. It's these hormonal environment has some effect there. Odds ratio are not huge, right? They're not a causal factor, but they are related in a way. If you make it more complicated, this is one data set from Craig Newshafer's study, the early cohort, which is also a high probability cohort. So these are kids who have an older sibling who have an autism diagnosis already. And they analyzed prenatal environment by some proxy. So here on the left, you can see that they analyzed the association between umbilical cord, which is at birth, testosterone level, and the child's behavior outcome associated with autism, measured by ALC or SRS scores. Now, over the whole cohort, you don't really see an association. If you stratify them by the proband sex, then you actually see some trend, or actually quite significant but small sample, associations with those kids who have an older female sibling. Now, the idea there was that those kids who have an older female sibling may have higher genetic background associated with autism. So the implication here is that if the prenatal environment is having a role, it might be interacting with the genetic background. Now, again, the question for all these is that this is gene-environment interaction, or even some correlation, and do we have the power to detect that? In the general population, we don't know. And this is, again, high probability cohort. But it does point to the probability of the joint effect of all these factors contributing to what we're seeing, basically the association of mechanisms of sex differentiation and mechanisms of autism. Now, at the human level, this is a work from, again, led by Dori Flores and Han Peng, fascinating work. So what in this study, we looked at the hypothesis of sex differentiation mechanisms may be related to the brain morphology. So the idea generally is that if sex differentiated mechanism somehow has a way to shape the structure of the brain of the person, then if you construct a sex label prediction model, then this model will work less accurately for autistic females, because you get some crossed effect of autism effect versus sex differentiation effect. It might work better for autistic males because that effect actually goes hand in hand in the same direction. So what we did, is that took the UK Biobank, large cohort, thousands of people, using a deep learning neural network to predict the sex label with the T1 weighted image as the input. With that, use that through transfer learning to the ABI data set. Now, resample the ABI data set by sex and diagnosis, balance sample for 100 times, predicting the sex label across all the data sets, and then you have a distribution of how accurate you actually get the sex label correct. The comparison is to what happens for the neurotypical sex-based prediction, which the differences should be 0 because that's what it's meant to work. It actually does work that way. What they found is that the sex label prediction model works poorer for autistic females compared to autistic males. It works poorer for autistic females compared to non-autistic females. It works better for autistic males compared to non-autistic males. Now, the same model trained by Abide has been used to predict what's happening in LEAP sample, which is a European sample, showing the same findings. It's not predicting the ADHD data set. So ADHD cohort doesn't show the same effect of this misclassification by sex, by the sex prediction model generated from UK Biobank. That differentiation of the prediction accuracy is more prominent for the younger age groups compared to the older ones. But it is not implying that autistic females have an extreme male brain. Because what's happening here is that if you look at the individual distribution or the prediction of the sex label of the individual's brain image, you see most female individuals assigned at birth are predicted to be female across the board. It's just that the distribution becomes more widespread, have a long tail for autistic females, and the same for the male population. So that's an interesting observation about what the convergence at the neuroanatomical level might be in terms of brain structure. And this paper is coming out in AJP, hopefully in a couple of weeks' time. So the final one I want to mention is from Donald Whirling's group, which is talking about what does it mean by that convergence in the molecular level. So Donald's conceptualization is that if the two mechanisms do converge, then it could be either direct or indirect. The direct one is sex differential mechanisms have an effect directly to autism-related mechanisms. Or it's actually the downstream effect of the convergence. So what Donald looked at is in a brain gene expression data set to look at how likely they converge. Donald does not find a convergence supporting that sex differentiation or sex differentially expressed genes has a direct effect to autism-related genes, but they do converge at the downstream aspect, especially the immunological and microglial pathways. So the idea, I think, that the implication is that there seems to be some convergence that's like neuroendocrine, but also immunological, and that may be related to metabolic as well, because we know the placenta, the fetus, and the maternal unit actually are affected by all these mechanisms altogether. So a quick recap. So we skimmed through a lot of things related to sex and gender. As you can see, we struggle because it's complicated, but we do have these angles or lens to look at clinical aspect from presentation to recognition, and sex and gender can serve as moderators. They all will have impact on delivery, although we're probably not there about what does it actually mean by a gender-informed or sex-informed clinical care. I think we're increasing awareness, but we're not quite there yet. Not to say about intervention outcomes, for example. But they also are related to being moderators or mediators of the emergence of autism. So we need more studies. This is a very nice summary of the published psychiatry and neuroscience studies, basically saying that, okay, what's happening 10 years ago between in 10 years' time. So as you can see, there's better use of both sexes now compared to 10 years ago. The optimal design to discover sex and gender effects are still, actually in this case, sex effect, are increasing, but still not there. Only 19% of the studies actually has that optimal design to look at. And the optimal design actually includes, do you have enough sample for you to look at those questions? Not to say those who can actually afford an optimal analysis to look at sex and gender effects, it's like 2% and 5% across 10 years' time. So we're not there. Sex is less omitted, that's good, but still, there's a problem. And that problem relates to equity. Because what happens is that we have a lens to include people. There could be people who are excluded from the outset because they're actually not considered autistic by whatever criteria that we use. And then, in the pipeline of studies, they also could be excluded because they have a co-occurring condition. they have epilepsy, they're non-speaking, they have cognitive impairment, so they're excluded from neuroimaging study, for example, to start with. And then you learn from those, you apply those knowledges, seeming that that's actually representative to the autistic population at large, which is not the case. And then you apply that back to this, and then people are excluded as well. Then you don't know, because it's actually a self-fulfilling prophecy about, this is what autism is, but you just don't have the data to speak to that. And I didn't put out this slide after coming here. I actually put this slide a few weeks back, which is a great study led by the Gabrielli lab, looking at what in practice it looks like when you apply a certain criteria to exclude people. And in this case, I believe, is using the ADOS criteria to see how many people you exclude. You see that in the male sample, you have 20% excluded. In the female sample at birth, assigned at birth, you have 50% excluded. So there's something happening there regarding how we decide on who to include. And I would argue it is important to understand or actually think about what are the purpose of our study, and then to decide on what inclusion criteria we're going to use, and then to get the knowledge that we want to get, but not to be too dogmatic or orthodox to say this is the only way that we can define our quote-unquote caseness. So I would like to thank all of you for inviting me, and I know I go over time, I always do. And also I want to thank all the autistic people and families that I talked to and the funders for the research. And I'm glad to have any questions to be discussed.
Thank you. Questions? Let's use the microphone so that people on YouTube can also.
Thank you. That's really interesting and a lot to unpack. Is this working?
I think so, yeah.
So you kind of touched on this a little bit, but I'm wondering, and I've always wondered, how you take into account the issue of intervention and how that kind of translates to how people present, right? Because intervention specifically for autism is also very often is very targeted at the type of behaviors that are then used in order to do the diagnoses. And so do you think that that might play a role also in the sex differences? I guess I don't know what the treatment differences, what we know about treatment differences in intervention.
Yeah, that's a great question that I don't know either. I think what we know is that we're all shaped by experiences, right? And as you said, interventions or treatment is a quite targeted way of experiences, especially how people are encouraged or prohibited from doing. Now, they definitely will shape our behavior. I think that's the case. So it's tricky, right? Because you want to observe autism-related behaviors in certain experimental setting, and then you have people who have received certain intervention for that not to be seen to start with. That's the whole goal of intervention, right? So, okay, so that's a bit too far. But I guess what I'm saying here is that I think all these intervention history, treatment histories can definitely have an impact on what we're seeing. Tricky part being that we just cannot control for all of them because they're so diverse. They happen in different timeline. I know a lot of researchers actually take a record of what kind of intervention has been received and trying to create variables to demonstrate that effect. I think that's a great way to do it, although we just don't have the, probably the most valid estimate to that. We don't know whether that's depending on sex and gender too much. I would suspect that they would be, in the sense of gender socialization does have an effect, in the general literature of shaping how one acts and respond. So I think that has a role. I think the question there is that we don't know how wide the variances is because of the variety there. So again, there's one ideal study I always want to haven't done is that how can we quantify gender socialization effect for the individual that we're looking at, apart from the individual self-report. So can we quantify that in a certain way to help us understand sex and gender impact on the outcome?
And I think that brings up something that also you touched on that could be a really good tool, which is a longitudinal individual changes.
Yeah.
That would be.
And I think that's why I personally really appreciate those longitudinal studies that has been there. As you might know, that there aren't that many longitudinal studies for autism, even nowadays. But there are this interesting like sex/gender, again, we don't know, like dependent or even like affected trajectory changes over time. I think Christine Unorda's study showed that, and also Peter Zamari's study showed that you do have more enriched girls who showed this declining ADOS score over time. So what that exactly means, I think Christine is actually collecting data for her wave 4 and 5 to look at, I cannot speak for her for sure, but I know that they are doing studies to look at what the experiences are into teenage. So maybe they'll be able to delineate what the developmental effects might look like.
Thank you.
Other questions?
Really great talk, thank you. So I'm curious, as a diagnostician, we're asked to contend with impact on functioning, and I'm curious how you think about masking for girls in the context of functioning.
Yeah, that's another great point. I think we're still building consensus on that. I think the empirical side, Sven Bold's study on the ICF actually has a paper outlining how professionals view the functioning aspects can be applied similarly or differently across the gender. I don't recall, I shouldn't speak for them, I don't recall that they have different, for example, threshold or scoring, but I think the finding was that users of that perceive, again, more of like internalizing challenges for girls and more externalizing challenges for boys, and then higher social function in general, but that's a perception from the user. I have two things to say about functioning. So one, we all rely on quantitative functioning assessment for characterizing that, which is a normed assessment according to age, which is a good start for us to get some general like perception or estimation of what it looks like. And if it is normed, it should take into account of the gender or sex effect of that by definition, if they do norm that. However, also, I think there are also other aspects of functioning that may not be captured by adaptive functioning itself. So, for example, quality of life metric is one key thing that people are developing. Like, what does it mean by having a good quality of life, given your neurodivergence? And I think that will and should take into account one's sex and gender effect. Although again, that's not my work, so I cannot speak to how much people have specifically looked into that. I do think that's something to be included, should be included.
Hi, thank you for that great talk. So I've been conducting some research on trying to find out how women who have autistic women with normal intellectual ability talk about their frustrations with clinicians and their frustrations with their daily lives. So one of the examples is the difficulty of getting a diagnosis. So I'm sure you know about some of this, but I know you haven't really talked about it. Things like, well, I couldn't get a diagnosis because a clinician said I had good back and forth conversation, or I'm married, or I'm doing well at school, or I have a relationship. So what these women talk about is having social problems that are real, that are far more subtle than the kind of stereotypical prototypical autism that I have. So eye contact is fine, subtle back and forth, conversation is fine, but they talk about having very subtle social problems, like difficulty with conflict, difficulty standing up in an interpersonal situation and arguing for themselves, self-promotion, difficulty, relationship difficulty. And so then all of this they end up with getting borderline personality diagnoses, or depression personalities. One quote from a woman has just really stuck with me, it's so powerful. She talks about waiting a year for her diagnosis, going in, the clinician's asking her questions about something that could be disturbing, and she ends up crying. And then he gives her a depression diagnosis. And she says, well, what she's having is a meltdown. But the meltdown isn't, writhing on the floor, it's basically crying. So I just wonder what you think about some of these very subtle social deficits that clinicians don't seem to know about.
Thank you. That's also what we have learned and heard from a wide range of autistic lived experiences and across different countries as well. And I think, yes, so the subtlety should be highlighted and made known for people, for clinicians, practitioners, and that's what we've been trying to do in terms of outlining the nuanced phenotype idea. There's a book that I refer to. It's actually by Donna Henderson and Sarah Wayland, two fantastic clinicians who actually wrote a book, was going to be published next year, about like the subtle autism presentation that learned from not only girls and women and gender diverse people, but like including. So yes, I think there are lots of awareness of that. Now, the more complex issue, two more complex issues. One is that, let's say the DSM criteria doesn't give you clear behavior examples of how to score it, right? Compared to the ADHD criteria, for example. So there's definitely the room for clinical judgment and decision making to say whether this fits. Now, whether this fits is actually reflecting the clinicians, almost like the gestalt they have in mind. And that is hopefully reflected well by the DSM criteria. It's not always the case. So people can, interpret in different ways. So for example, I would not count back and forth eye contact to say there's no nonverbal communication. I would have to know what that eye contact looks like, what does it look like over time. And that is what I meant by this epistemic iteration of change of diagnostic criteria, because we can see actually from DSM-4 to 5, this has been highlighted quite well, and you also see that from DSM-5 to DSM-5-TR, that has been highlighted. So for example, the presence of characteristics either historically or currently is put in there. So that's kind of reminding clinicians, things can change. So as long as you have data from somewhere in the life, and as long as the overall picture has a clinical implication, you should actually use it as your diagnostic evidence to support your finding. The DSM-5-TR actually has a bit more talking about these like developmental changes, especially the ideas of like masking, camouflaging, whatsoever people call it, in the text, not the criteria itself. So research has gone into the diagnostic manual, but you're right that how people perceive it, how people read it and operationalize it, I think that requires lots of conversation. That does involve shifting or the norms diagnostic concept, which may be reflected in the criteria iteration moving forward. But it's a gradual process. It's also a negotiation process. Like, how would a clinician be persuaded that I'm comfortable with this is autism instead of borderline personality? That's a conversation that has to happen between personality, psychologist, psychiatrist versus neurodevelopmental psychiatrist, psychologist. And I think that level of conversation needs to happen. So I think it's a gradual process that we all learn. And I'm an optimistic person, so I think that's what I see it.
Wonderful talk. Thank you for coming to MIT. I was wondering, so one of the things that we've seen at MIT in our characterization of participants is relatively lower scores on the ADOS in women consistent with the literature, very low levels of repetitive behaviors, but elevated scores in self-report measures.
Yeah.
And I wonder if you could comment on the role of self-awareness and other related factors like alexithymia in how you see the conversation about sex differences and other differences evolving and how we study it.
Yeah, thank you. That's another hugely important question. And okay, so I think the role of self-report measure, it's a double-sided sword. Because most of self-report measures, if you look at psychometrics, they're not diagnostic measure by the standard that we used to have for a variety of reasons. But on the other hand, they are self-reported measures, meaning that they can actually help us have access to the information that you don't get traditionally as a neurodevelopmental practitioner. Because traditionally, the field relies on behavior observation and parental report. Right? These are like the key thing that we rely on for children, and understandably. But I think the role of self-report is I would argue is an integral part for assessment if we can get that, right? And then I think, and again, DSM says that actually explicitly. You should have that if you have access to. Now, so I think I'll definitely take into account of those self-reported measures. Again, the other issue is that self-awareness is also important one. So for sure, you may have higher self-awareness, so you have higher reported scores. And some people may actually argue against the use of the screening measure because you may not actually have higher self-awareness, so you think everything is fine, so you score low on that. So it can act either way. And I think the clinician's role is to have a holistic judgment to see, okay, are they going consistent direction? Right, so if my resource goes diverging directions, I have to make sense why they go diverging directions, and that's often what we do with harmonizing school report versus parent report versus self-report, and that's clinicians' work. So I'm not sure if that helps with the
So I kind of have a follow-up question to that because you're talking a lot about the clinicians and how it can be clinician dependent and so on, and thank you. So all of these studies, they talk about studying the person or the child themselves. What about studying clinicians, right? Like in trying, double-blinded way to see whether there are any biases.
That's exactly, I think, what can and should happen. I think I cited one study with an experimental one to the school, primary school educators. So that's not clinicians, but they're educators. That's one piece of evidence. There's another study from the Australian group actually asked the clinicians' rating given vignettes, and then asking about their confidence of diagnosing. What is interesting in that Australian study is that clinicians actually are not biased to underdiagnose autistic females given that vignettes. They tend to actually have given higher rates of higher scorings based on DSM scores for autistic women if presented in vignettes. So the manipulation is still the name of the implied gender of the vignettes. But the clinicians are less self-confident in diagnosing them. So the Australian study actually gives you a different direction compared to the clinical study. The educator study that I cited in the UK. And we don't have other study that I know of so far. I think it's discipline dependent, it's region dependent, and
Well, and nowadays it's also, there are a lot of clinicians, especially when they're thinking about more like high functioning, that very openly become more flexible depending on what the goal is. So sometimes the goal is just to provide some kind of support, and this is, diagnosis would be the best way to provide that support. And so that also would change how they.
I think we have a lot of variability in terms of how a person gets diagnosis in the end. I mean, to one extreme, you have like huge difficulty getting. On the other hand, there could be overdiagnosis that's happening at the time because of higher awareness. And again, depending on how rigorous the assessment and the whole process goes. So we're in a quite muddy space now. Like there's a quite wide variability, like from under awareness to, I wouldn't say over awareness, but like
Yeah,
hyper.
I don't necessarily think it's overdiagnosis, but yeah.
Yeah.
I just mean that like you said, different clinicians in different places and different settings have
Yeah.
different goals.
So given the time, let me ask one final question from the YouTube audience. This comes from Vishaka Shukla, somebody we know. So Vishaka asks, comparing female presentation across different race identities might help tease apart the merely biological effect from socialization kinds of effects. What do you think of this possibility?
I think it's much needed. I think it's definitely possible because you're now, in a way, like controlling for sex, and then you look at what the gender-related experiences might be. So I think one ideal research design, if you want to tease sex and gender effect apart, is that you first of all you have enough stratification, you have enough sample to start with, and then you measure both, and your analysis should actually be within sex or within gender analysis. So you actually sort of like you don't control that in the overall model, but you look at it separately, and then so you'll be able to look at what the effects are within your strata. So yeah, I think that's a good direction for sure.
Great. Thank you again very much for the wonderful question.